I'm Bjorquist, CEO of Verigraft from Sweden, the other side of Europe. And life is a bit upside down because yesterday when I left Sweden, we had 22 degrees Celsius and sun. And when I came to Spain, it was 15 degrees and rain. How can that happen? Must be once in 10 years. Anyway, let's not talk about the weather. Let's talk about Verigraft. We are a small classical university spin-out company from the Karolinska Institute in Stockholm. We are now with the headquarters on the other side of Sweden, being Gothenburg on the west coast. We are privately funded. We have owners from Sweden, Switzerland, Korea, and the US. So we are really a global company, although small. We have a highly experienced management team, and you can see us here. And to cut a long story short, we have experienced about 20 years from regenerative medicine field, and we have grown companies before in this area. So what is our business model about? So we are doing tissue engineering, and we are making transplants personalized. And I will try to tell you how. So what is all this about? Coming from Sweden, I have to show you a lobster. And this is, of course, a lobster, but you can see that this lobster is having one big claw and one small claw. And that is what all is about. A lobster can regenerate a claw. We cannot regenerate an arm or an organ. So we have to help the nature. So we sometimes see these type of pictures. And to cut another long story short, we are not ready really to produce organs de novo with bioreactors or any other equipment. So we are not doing that. We are not creating organs. We are helping the patients by personalizing organs and make them useful for transplantation. So again, I have to tell you how. So this is a donor, a cadaveric donor, donating a tissue, in this case, a blood vessel. Note, in the future, these donors can also be animals, so we think we can also use xenogenic material in the future. We anonymize this blood vessel by a process where we take off all what makes the organ or tissue to be specific for a patient, being the cells and the DNA. So we end up with a scaffold that is basically a rough protein tube in this case, but still with exactly the same three-dimensional structure. Make another note here. We can easily freeze these tissues down. So a scaffold with these proteins can easily be stored in our freezers. Next, and importantly, we are taking material from the patient, in our case, blood. So we are taking whole blood, we are adding some tricks to the blood, and we are soaking the structures for only 1 week. And by this incubation with blood and our specific tricks, we make the tissue, in this case the blood vessel, ready to be transplanted and with a new identity belonging to the patient. And of course, we are doing this because we want to bring an allogenic material into something that is found to be autologous by the patient. So this is all about to avoid the requirement of immunosuppressive agents. So we should cut a long story short of several years of preclinical testing. So this is a pig model from Sweden where the surgeons at the big hospital in Gothenburg are replacing a vein in a pig with a personalized vein. And you can see on the bottom two pictures that there is a very good blood flow in this new blood vessel after a while. And you can also see that the difference between the PTEV, that stands for the personalized tissue engineered vein, and the surrounding tissue is almost not possible to identify. You can probably, if it's dark enough, see it even better here. On the top level, you have the native vena cava from these pigs, and on the bottom row, you have our personalized tissue engineered vein. And you can see that the amount of cells are almost the same after operation and a certain period. And you can also see that the cell types, most importantly, the endothelial markers are there again. So what are we heading for? We are heading for treating patients, and our first indication is called chronic venous insufficiency. It's a blood vessel in the thigh region, and importantly and interestingly, these veins are carrying valves that are opening and closing and allowing the blood to go back to the heart, but not in a too extensive manner, go down to the leg again. And as you see, in some patients, these valves are destroyed, and then the patients are starting to get pain and swelling of the legs, but unfortunately also ending up with leg ulcers. So we are producing a piece of a vein with functional valves. So this is our product, and you see a piece long enough to carry a pair of valves and to be sutured into the patient by the surgeons. So we are heading for a first 1/2 trial in Europe. 10 to 15 patients is only a small study. It's classical replacement surgery, etc. And we are going to study, of course, safety very carefully in these patients for 3 and 6 and 12 months. So the entire study is 12 months. And we also hope to see signs of efficacy. So this is a unique trial, of course, first of its kind. So we are working intensively in Europe right now to find the patients, to find surgeons, to find labs where we can produce the material. So we are truly collaborative. We are working in some key countries for us. So we have very strong collaborators in Lithuania. You can see 2 of the companies, Froset and Clinical Accelerator. We are here in Spain, which is of course interesting, and we are working very closely with Andalusian Initiative for Advanced Therapies, and we also work in Switzerland. So we are fully focused on bringing this to clinical reality. We think for the future that, of course, now with US owners, we will bring this to the US market. After Europe and in the rest of the world, we are looking for license agreements. So, looking specifically for a few seconds about this chronic venous insufficiency, it's a big disease. It's more than 1 million suffering only in Europe and the US from this disease. And with no treatment today at all for these patients, it's a virgin market. And with a strong reimbursement model we have and low costs and cost-efficient production, we hope to target a €1 billion market in the future. Just to mention that this is only the start of a very fantastic journey, we believe. So we are now working in preclinical models, in large animal models with arteries. Together with collaborators, we are also creating nerve, the peripheral nerve that also now are tested in large animal models, actually in Spain as well. So this is part of the future, just to show you the potential of the technology, and that we are addressing several very big markets with the technology. So as a summary now, we are here because we are very collaborative. We would like to find more partners for our technology. We are, of course, looking to out-license, as I mentioned, the technologies. We are looking for that. And we will soon start a new fundraising. And just to pinpoint that a little bit, the strength of this proposal for potential investors, we have a very experienced team. We have worldwide granted patents that are very strong. And on top of that, we add a layer of trade secrets, making it even stronger. We have a European clinical network that is strong. We have relatively recently settled an investment round, and we are looking for a Series C round now, targeting second half of 2020. So it soon start to work very hard on that one. So since we are collaborative, we have a lot of money. I would like to honor our funding bodies here in this slide. And to sum up, and tell you that I'm happy to be here. I'm happy to share these data because we really want to generate not only the lobster but also human beings. Thank you very much for listening.